So in the rest of this chapter, we're going to look at osmosis and also at something called bulk transport. So just to review, we've gone over simple diffusion. There aren't very many molecules that can go directly across a membrane, but oxygen is one that can, and carbon dioxide is one that can. Pretty much everything else has to either go through some kind of channel protein, carrier protein pump, which is really just a carrier protein that does active transport, or it can go through a vesicle by phagocytosis or exocytosis. So we'll go over those things. All right, but let's look at water. This is really water going across a membrane. Water always has to go across a membrane through either a channel protein or through bulk transport. So we're going to look at it mainly through a channel protein in this section. So osmosis is the diffusion of water across a membrane. So when you talk about osmosis, you are literally only talking about the movement of water along its concentration gradient across a membrane. So we've been talking about other solutes. Solutes are things that are dissolved in water. So sodium, we talked about sodium moving across a membrane. Potassium, those would be solutes. Glucose can move across a membrane. What other examples did we have? But the solvent is the liquid, and that's always water for a living organism. So water is the solvent. So osmosis is how the solvent, how the water diffuses across a membrane. It follows the same rules. It's just that you have to figure out the concentration of water in a solution. And that's tricky because concentrations are always given from the perspective of the solute concentration. They're really never given from the perspective of the solvent. So you have to, in a way, think kind of in reverse about it. So this is a kind of a classic experiment. This is set up in a beaker, or there is an open glass tube that can form a U shape. So it's literally called a U tube. Let me get my pen, but U tube. In this picture, though, they just did a glass beaker. And in the middle, you put a membrane. And in this case, it's going to be like a plastic membrane. It's not going to be a phospholipid bilayer. But this is like a piece of plastic meant to mimic the way a membrane would act. It has pores in it, but the pores are a certain size. In this case, the pores are too small. These holes are too small for the purple solute to go through. So if the solute cannot pass through the hole, for example, in this example, and you'd have to be told that, what can pass through the membrane, what cannot, if you had a sort of a theoretical situation like this one. And the solvent, which is just shown in pink, I don't know why they chose pink, but this is the water. All right. So whatever the solute is, it's not able to pass over the membrane, but let's say that the water can pass through these pores in this membrane. What's going to happen is if you look at these two, like this side of the beaker is sort of, let's call it side A, and this side is B. So you literally pour two different solutions on either side of this separating membrane, because it completely separates the beaker into two parts. And what you'll notice is that solution A is pretty diluted. It doesn't have very many solutes. So the solute concentration is pretty low compared to solution B. So the solute is very high on solution B and very low on solution A. So if the purple molecule or the purple solute, if it could go across the membrane, it would go this way, right? It would go from right to left because solute is going to diffuse down its own concentration gradient. So its area of high concentration, which is solution B right now, to its area of lower concentration, which is solution A. But that's not going to happen because the solute can't move. But the water can move, and the way the water is going to move is going to be the opposite direction, because if you look at solution A, and you can kind of notice, you can think of these as being exactly the same volume. A and B are the exact same amount of solution. The amount of water in A is higher than the amount of water in B. And that's because in B, some of that space is being taken up by solute, and therefore there's less solvent in B. So A has less solute, more solvent. And B has more solute and less solvent. So you want to think about each of these in comparison to each other. This is always in a comparison. You know, what would you say about the solution on one side of the membrane compared to the other side? So solution B has more solute and less solvent. So what we would end up with is, if water is the only thing that can move, it would always move from its area of higher concentration. So more solvent is A, more water inside A, and less solvent on side B. So the water would move from left to right. So the purple thing would move from right to left if it could, and the water would move from left to right if it could, and the only thing that can move is the water. So the water will move, and what will happen later, so this arrow means time passes. In this case, the arrow does not mean that bonds are forming or breaking. It just means time passes. And so after the water moves from solution A into solution B, only the water, that would be osmosis, because the water is moving from its area of higher concentration of water to its area of lower concentration of water. And what would happen literally is the volume, or the height, I guess you could say, of the liquid on side B would actually go up. And this actually works, and it looks really odd, but it will work if you set this up in a lab situation. Now there is a point where when the water leaves solution A becomes more concentrated with solute, and there's a point where the two sides become equally concentrated and the water then stops moving. There's also a point where gravity will prevent this side B from being able to get higher. But in a theoretical situation, it would only stop in the sense when the two solutions are equal concentration. But that's how you figure out which way the water will move. I would think about where, when you're comparing two solutions that are separated by a membrane, think about what's the concentration of the solute on each side of the membrane, what's the concentration then of the solvent on each side of the membrane. Because if there's a high amount of solute, that means there's less amount of solvent, and if there's less amount of solute, there's more solvent, so more water. So then you can figure out which direction it will go. The solute and the solvent will always want to go in opposite directions according to diffusion. So we have terms that we use to compare what we call the osmotic concentrations of a solution. A hypertonic solution is the one that has the higher solute concentration, compared to the hypotonic solution is the one that has the lower solute concentration. And again, this is written according to the solute, not the solvent. You have to use these terms in comparison. You can't just have a hypertonic solution, period. it has to be A is hypertonic to B or B is hypertonic to A. In this example we had here, B had a higher solute concentration, so B is hypertonic compared to A. And then, or you could say it the other way, you could say A is hypotonic compared to B. But you always have to have a compared to statement. The only other possibility is if the two solutions have equal concentrations, we call them isotonic. So isotonic would be the end point. That would be when the two solutions finally reach equal concentration, osmosis will stop, and the two solutions are called isotonic. It's really the end point. That's when osmosis would stop. Now, like I said, only oxygen and carbon dioxide can cross a membrane, a biological membrane, directly. And so in the membrane, in the plasma membrane, there is a channel protein called aquaporin, which you probably know. Aqua is Latin. It's from the Latin root for water, and you can see the word pore. So it's a pore for water. It's a channel protein that lets the water go according to its diffusion. So it's facilitated diffusion through the aquaporin. So only water can go through aquaporin. All right, now, so what would happen if, let's say, water is going into a cell by osmosis? Let's say there's more water outside the cell in terms of concentration compared to the inside of the cell. There's going to be a point where the water is either going to pop, like burst, or somehow it's got to stop. So if we're talking about a cell that just has a cell membrane and doesn't have a cell wall, if osmosis keeps moving water into the cell will burst. But for cells that have a cell wall, eventually the cell wall will hold the cell together, and the cell will become so rigid because it's so full of water that even if more water tries to come in, it's pushed back. And that's called osmotic pressure. All right. Actually, the osmotic pressure is the force moving into the cell, but then the cell wall, because of the cell being so full of water, it counteracts the osmotic pressure. All right, so only animal cells have the problem of bursting, actually. I think I mentioned before, animal cells are unusual because we don't have cell walls. All other cell types, except for some protists, but most other cell types have it. Bacteria have it, fungi have it, plants have it, a lot of the protists, not amoeba, but a lot of the protists do have it. So it's, it's a pretty small group that doesn't have it, and really having a cell wall is going to be important if you're in a freshwater environment. So let's look at what would happen if you took an animal cell, and we're going to use a red blood cell as a typical animal cell. And this is kind of an example that's used. You'll see this in other courses, and you'll see it on standardized tests if you take an entrance exam for a medical school, or, they always love to put cells into different solutions, and you have to know what's going to happen. You have to know what the outcome is. So if you put a red blood cell into a solution that's hypertonic, now these words up here are saying the solution is hypertonic, and so by definition, what's inside the cell would be, by comparison, hypotonic. So the solution in the beaker, let's imagine a beaker, the solution is hypertonic in this first one. And then the cell is inside the beaker, and the inside of the cell is hypotonic. All right. So the inside of a cell typically has about, 0.9% saline. So that's usually a good kind of number to use. 0.9% saline. That's 0.9%, so just slightly less than 1%. So 0.9% saline here inside the cell. So a hypertonic solution would have much more of the saline is sodium chloride. So this one, you know, maybe 20% salt outside the cell, outside in the beaker. So if you dropped a cell that had 0.9% sodium chloride into a beaker that had 20%, I just made up that number, then which way would the water move? You have to think about this because we're only looking at which way the water is going to move because we're talking about osmosis. So where is the water concentration the highest? 20% salt solution is very salty. 0.9% salt solution is more diluted. So the more diluted one has more water. So in this case, the inside of the cell has more water. I'm going to put an M for more water. It's just for the water. And the outside of the cell has less water. And water is going to move according to its gradient from an area of more to an area of less, so water will come out, and that's what they drew here. But I'm giving you kind of made up numbers, and you can then go through that logic process to think about that. In a hypotonic solution, what that would mean is if you put a cell into a solution, and the cell, let's say, has 0.9% still saline, but maybe the solution is 0.1% in the beaker. So where is there more water between these two solutions? The beaker solution actually has more water because it's more diluted than what's inside the cell, less water inside the cell. Water will move from its area of more to its area of less, so water will move into the cell. It depends how much of a differential there is between the two solutions. If the water moves in very fast, it can make the cells burst. When the water makes the cell burst, we call this lysis, the bursting of the cell, lysis, L-Y-S-I-S. On the earlier example, when we put the cell into the hypertonic solution and the water left the cell will kind of crumple up and shrink, and we call that crenation. Not cremation, crenation. That is just the cell shriveling up. Kind of what happens when you go into the ocean. Your fingers wrinkle up. Okay, because it's salt water. Okay, anyway. That's not really why they wrinkle up, but you can think of it that way. Crenation is when the cells shrivel up because all their water is going out through the aquaporins. The cell isn't destroyed though. Lysis though kills the cell. So lysis kills the cell. Crenation, it's not the greatest, but the cell doesn't die. But lysis is death for the cell. So the cell will either swell, well, the cell will definitely swell in a hypotonic solution. If it doesn't reach if it doesn't reach isotonicity, it will keep swelling until it bursts. Isotonicity is when you put a cell, if you put a red blood cell into a beaker, and the beaker had 0.9% saline and the beaker had 0.9% and the cell had 0.9%, then that would be an isotonic solution and you wouldn't have any water movement. Or what we say is you don't have any net water movement. So if some water goes in, some would also go out and it would cancel out. Blood cells are most happy, I'm going to write happy, they're most happy in the isotonic solution. They die in the hypotonic, if it's very hypotonic, or it's not good. They're not too happy in hypertonic, so the best scenario is isotonic. So when you go, if you have dehydration and you go to the hospital and they put an IV in to give you fluids, they don't just give you water, because if they filled your veins with water, your blood would become hypotonic and then it would make your cells burst. Really all your cells. So they give you a saline solution in the IV because they're trying to match what's already in your body, isotonic. Now, what happens with plant cells? The direction is the same. the outcome is a little different. If you put a plant cell in a hypertonic solution, just like before, the water comes out of the cell. However, the cell membrane is attached to a cell wall, and the cell wall stays in place. So the cell membrane shrinks up. We don't call this crenation, though. In the plant kingdom, we call this plasmolysis. And I really want to apologize because the word lysis is in this one. But this is plasmolysis. It's the shrinking. The reason it has the word lysis in it is that the cell membrane is splitting away from the cell wall. So in that sense, there is some splitting. But it's not the cell really splitting or bursting. It's the cell wall shrinking inward. And so this is the equivalent of crenation, but plasmolysis is the word you use for a plant cell. In an isotonic solution, really the cell is what we call flaccid, kind of floppy. It's okay. But the happiest scenario for a plant cell is to be placed into a hypotonic solution where the water will rush in. And I didn't point it out earlier, but there is what's called the central vacuole in the plant cell. It's the largest organelle in the plant cell. And it's one of its main purposes is to hold water to keep the cell rigid. And plant cells like to be rigid because that's how they stand upright. It's part of how they stand upright. Is when a plant takes in fresh water, it will fill up its vacuoles and it makes all the cells become full. It puts pressure on the cell wall and that helps the cell stand upright. We call this pressure turgor pressure, or we say the cell is turgid. Turgor pressure. So here's what happens. I think you know what happens when you don't water a plant. Among other things, the cells become flaccid, and that's what you see on the left side, and limp. On the right side, you see when you water the plant, the cells are full of water, the cell stands more upright, it's more, leaves are spread out, and that's because of the turgor pressure. Now, for organisms that live in fresh water, especially single-celled organisms that live in fresh water, it's a constant problem because if you think about it, if you live in fresh water, the water's going to flow through into the cell constantly. And for something like a paramecium, even though it does have a more rigid structure on the cell, it has to have a way of getting rid of all that water. So it has, I kid you not, and I'll try to find the YouTube video that I have of this, but it has these little structures, they're called contractile vacuoles, and they fill with water, and then there's a little tube, I guess you could say, that goes to the outside of the cell, and they will contract and push the water out. It's the equivalent of, you know, like let's say you're in a boat that's sinking and you're using a bucket to dump the water out. It's like that. The water keeps flowing into the cell and the cell it fills up these vacuoles and then contracts them and it squirts out. I guess squirts. It's all underwater, but it pushes it out. It's really interesting, and that's the only way it can live in fresh water. All right, our last topic, bulk transport. We talked about this a little bit in, really quite a bit in chapter 4, because one of the things I need you to know about is how cells bring things in. So they can either come in directly by simple diffusion, but that would only be really oxygen and CO2. Then they can come in through a channel protein or a pump or carrier protein. And then the last way that they can come in is through bulk transport. So bulk transport is moving things into the cell by a vesicle, or releasing things out of the cell by a vesicle. So it's always going to be a vesicle either forming to move something in, or a vesicle fusing with the plasma membrane to push something out. So you'll remember that we talked about two types of exocytosis. Secretion is when the cell makes something good, like a protein, and it sends it from the ER to the Golgi, and then from the Golgi out to the plasma membrane, and then the vesicle fuses with the plasma membrane, and that dumps the substance out of the cell. So that's a type of exocytosis. Excretion is when waste, things that can't be used, are put into a vesicle, and that's fused to the membrane, and those things are dumped out. So excretion is related to waste release. Secretion is related to the release of something good and useful. It just needs to leave the cell in order to do its function. So endocytosis, how things come in, is something we talked about a little bit. Phagocytosis is how a cell brings in solids. It's really the fundamental way that cells bring things in through bulk transport. And that's why I underlined it and put it in boldface. Pinocytosis, the only time I've ever heard of pinocytosis is teaching this class. Like, I never came across it in any other way. I guess it could be a controlled way. The way I understand it, typically explained, is that it would be a controlled way to bring in water rather than through the aquaporins. But honestly, it's not super critical. Receptor-mediated endocytosis is really kind of a specialized kind of phagocytosis. So this one's kind of related, actually. It's just given a different name. Phagocytosis is where the cell sort of forms a vesicle around some material that's right outside of it, and that way it brings it in by a vesicle. Receptor-mediated endocytosis is where the cell puts receptors on the cell membrane, and those receptors catch things in the environment, specific things, and then brings it into the vesicle. So in receptor-mediated endocytosis, the cell is intentionally catching only certain things that it wants. So it's fishing, I guess you could say, for certain molecules. Phagocytosis is more associated with just kind of randomly bringing in whatever's there. But they're pretty similar in a lot of ways. And like I said, pinocytosis is not super important. Let me show you the phagocytosis, though. Let's say you have some kind of large particle, and it could, and this is kind of like time lapse here. At the top, you have the particle has to come pretty close to the cell membrane. And by the way, this only works for cells that don't have a cell wall. So that pretty much limits you to like amoeba, which is a protist, things along that line, and then animal cells. Plant cells can't do it because of the cell wall. Fungus can't do it. So phagocytosis is not every type of cell can do it. But anyway, for the cells that can do it, it's because their cell membrane is not attached to a cell wall that it's able to sort of sink down and sort of surround, and the membrane sort of flows around, and then this vesicle pinches off and moves into the cell. You can get a better picture over here on this other figure where it just moves in and then forms a vesicle, and the vesicle moves in. And then we usually call this, in phagocytosis, whatever the food is brought in, we usually call this a food vacuole instead of a vesicle. But vesicle and vacuole are fundamentally the same thing. So the food vacuole then fuses with a lysosome. So this should be a review for you. Remember, the lysosome is full of hydrolytic enzymes. We call it the primary lysosome. And when it fuses with the food vacuole, it mixes the hydrolytic enzymes with the macromolecules of the food. And it creates these, looks like hurricanes inside the cell, inside the what's called the secondary lysosome. And those hurricanes are really those hydrolytic reactions breaking down those macromolecules. And then anything that can't be digested stays inside the vesicle, stays inside the secondary lysosome. and eventually gets returned to the membrane for excretion. This picture is showing receptor-mediated endocytosis, which, like I said, is basically where the cell has these little receptors on its surface, and let's say it wants to catch the star molecule, whatever that is, and it doesn't want the square molecule. So what it does is the receptors will catch just the one type of molecule that it wants, and when all the receptors have caught something, then that activates the endocytosis event, the bringing in of that item. And this is, has special significance, especially in Dallas, because this is how the body takes cholesterol out of the blood. And the scientists who won the Nobel Prize for figuring this out work at UT Southwestern, Brown and Goldstein. And so anyway, they're in every bio book that I've ever seen. But this was their work. It's pretty famous. Exocytosis, like I said, could describe any case where a vesicle fuses with a membrane. In this case, the movement is going in this direction. And then that fusion dumps out the either wastes. If it's waste, then it's called excretion. And if it's some kind of useful protein, it's called secretion. But the word exocytosis really just means any scenario where something's released from the cell in this manner. All right, so endocytosis of any type, exocytosis, these are called bulk transport. And, larger amounts all at once, I guess you could say, is being transported across the membrane, either by formation of a vesicle or fusion of a vesicle to the plasma membrane for release.